Okay. Good afternoon. This is Nelly Poyak from Dana-Farber. Welcome to the DFHCC Connect Science Seminar Series. A reminder that the talk will be posted on the slides, will be posted on the website that you see on the first slide. And also a reminder to please post your questions in a Q&A function in Zoom, and we will answer as many questions live as we can. And ones that we don't have time, the speaker will have access to it, and we'll get back to you as well. And it's my pleasure to introduce today's speaker, Michael Angelo, who's an assistant professor of pathology at Stanford University. And he's been a pioneer of using the MIBI technology, the multiplex ion beam imaging, for studying tumor samples, particularly triple-negative breast cancer, that he will also describe today. And the title of his talk is Relating Single-Cell Function to Tissue Structure in Human Health and Disease Using Multiplex Ion Beam Imaging, or MIBI. Michael, thank you for joining us today, and we're looking forward to your talk.
Yep, my pleasure. So thank you all for having me. I am really happy to be able to talk to the people at all of you at Dana-Farber, I guess everywhere now, since we're all in a teleconference. So I'm still working on getting my Zoom presentation skills honed. So if I'm a little stilted, please forgive me. I'm used to looking at people in the audience. So my lab, we're sort of really, I think, interested in understanding sort of the nested organization of tissues at the level of single cells on up. And so what that means, sort of in a nutshell, is if we think about tissues being comprised of different cell types. So here, you know, if we've got like a fibroblast and a tissue macrophage and sort of columnar epithelium, when you assemble these into a multicellular structure like you find in tissue, how does the nature of the cell-cell proximities and how that structure is put together sort of drive the function of cells at a single cell level, and how does the function of single cells sort of feed back on the multicellular structure? So we really are trying to get at this interdependence between single cell function and sort of higher levels of tissue organization. And I think that's kind of the first aspect. And the second aspect is to try to compare how these sort of motifs are, or sort of, you know, eigenstates, tissue organization, compare across different scenarios, whether it's in health or disease. And so, you know, because we're really interested in understanding this at the level of, can you guys still hear me? I just got a warning.
Yeah.
Okay, sorry.
I can hear you. It's the subtitles that looks like disappeared, but that's fine. Okay.
So in order to do this, we really wanted to be able to do this directly in human tissue. And so in order to do that, you know, you need a method that'll allow you to determine single cell function. And really to do that, you know, the best sort of, the best way to do that is to, at this stage, is to be able to measure many different proteins at the same time within a single tissue section, while maintaining a high degree of sensitivity, a wide dynamic range, subcellular resolution. And for us, because we're primarily interested in doing this in human tissue, it's very important for us that we be able to have this method be back compatible with archival formalin-fixed paraffin-embedded tissue blocks, because by doing so, we're able to actually access virtually any sort of disease state or tissue type that we want by just dipping into, you know, a handful of the more than 1 billion different tissue blocks that are present in clinical repositories worldwide. And the other upside of this, right, is that because these were used in clinical care, we're able to also tap all the years of clinical annotation that go along with that. So sort of up until the time when we were working on using a mass spectrometry-based approach, really the conventional approaches for tissue imaging employ different types of immunofluorescence. So immunofluorescence, just kind of in a nutshell, if this is a piece of tissue that you wanted to image and you wanted to look at the distribution of different proteins, you'd have 3 different antibodies, each of them specific for the protein that you want to visualize, and each antibody would have a fluorophore tag to it. And so then the tissue would be stained with those antibodies, and upon excitation with the fluorescence with the light source, you get fluorescence emission, essentially telling you where the spatial location of these different proteins are. Now, immunofluorescence is really great. It just becomes harder to multiplex higher numbers of channels once you get beyond, like, say, 3 or 4. And that's because the emission spectra of the fluorophores start to overlap. And so as the fluorophores overlap, it becomes harder to really be able to tease out if the light's coming from, which antibody the light is actually coming from. And so one way to get around this is to move away from using light as a reporter and instead use mass as a reporter. So this is the idea behind both MIBI and also around mass cytometry or CyTOF, which is if you take an antibody and you tag it with a metal chelator tag. And so this chelator tag, it's got, for each antibody, you attach somewhere between like 80 to 100 different metal atoms. And the tag for each antibody has a unique mass associated with it. And so the benefit of that is that if you can image these mass tags in tissue, if you were to look at a mass spectrum as compared to like the fluorescent spectrum, the mass spectrum is far less constrained because you have adjacent mass reporters, and mass spectrum really don't overlap a whole lot. And so what that means is that we can go and we can do many more reporters, antibodies simultaneously without having a significant degree of crosstalk. And the other benefit too is because this is not an optical based technique, you don't have to contend with autofluorescence, which can be for multiplexed ion beam imaging by time of flight, is that we take the tissue section. We almost everything we do right now is with FFPE tissue. It's mounted on a conductive slide, and then we stain it with a single master mix of all of our primary antibodies, and each one of those primary antibodies is tagged with one of these metal reporters, and it's such that no antibody is tagged with the same mass. So every primary antibody has a metal reporter with a unique mass. The sample's then washed and dried out, and it's put into a vacuum chamber where the image is raster scanned with a primary ion beam. So this ion gun produces charged ions, whether it's xenon or gold or anything like that, that essentially are focused down to a really small spot size on the order of anywhere from like 200 nanometers up to a micron. And that beam then goes pixel by pixel across the image. And as it sits on a single pixel, we liberate secondary elemental ions. And in that elemental cloud, you have the antibody reporters themselves. And using time of flight mass spec, we're essentially able to see for every single pixel, we can see what the abundance of each one of our metal reporters was. And so, then with this data set that we have, where for each pixel, we have a spectrum that tells us the abundance of every antibody in each pixel, we can then go and extract that into spatial information. And so here, this is just showing, you can make all kinds of 3-color overlays using the data that's in that mass spectra. So, as I mentioned before, the goal of this is to try to be able to use the high degree of multiplexing that we have to be able to simultaneously both see the structure, organization, and lineage and functional phenotypes of the cells in the tissue. And so we do a lot of work looking, probably about half the stuff that my lab does is focused on some aspect of cancer, a lot of cancer immunology questions, but then the other half is also looking non-oncogenic diseases and normative physiologic states as well. And so we have that leads to like a a daily a pretty large flux of images on a daily basis. So we have about 12 active human tissue cohorts that are being sort of either optimized or validated and run at any given time. Most of the images that come out is around 35 plex data. On a given day, we're generating usually somewhere between 25 and 100 images. Each one of those images, those images in total essentially have around, up to 100,000 cells. So that ends up being kind of like a very rich and diverse data set that will that needs to be matched with pretty robust automated computational tools. And just to kind of give you a window into some of that complexity that we're trying to extract, this is a tissue cohort that we finished a few months ago. Shirley Greenbaum and Aaron Soon worked on this. It's been really a really fascinating study where we're looking at the immune cell subsets that are involved in maternal fetal tolerance in the first 20 weeks of human gestation. And so this was 70 patients, and we had 251 millimeter tissue cores that we analyzed in this. So this is a field from one of those cores. And so the first thing to see is just purely on a on a morphologic basis. If we look at just the nuclear channel and this little inset that I've boxed out here, you can see that we've got large cells, we've got small elongated nuclei, and then in terms of cell lineages, in purple here, this is HLA-G, so this is marking infiltrating extravillous trophoblasts, which are fetally derived. And so this is maternal decidua. So these cells are actually infiltrating into maternal decidua, much like tumor cells do. And those are surrounded and juxtaposed very closely by these maternal decidual cells, which have these long filamentous projections. But then we also have, in addition to these sort of stromal elements, you can see that there's these large open circles in there, which are lumen for blood vessels. And so in the bottom part of the screen, CD31, you can see is highlighting the endothelial layer of an artery that's surrounded by this layer of smooth muscle that during these first few weeks is actively being remodeled and disrupted. And so then there's a very huge diversity of immune cells that are present in the tissue as well, and they're all spatially very compact on top of one another. So here with CD45, we can see all the immune cells, and if we want to break it down by lineage, the three major lineages of immune cells that we find in there are NK cells, T cells, and monos. And so just to look at one of those, we had on the order of, I think, of like 8 or 10 myeloid markers in there to sort of be able to stratify myeloid cell subsets. And you can see that just looking at the myeloid compartment, with 3 of those markers, there's this really rich patterns of co-expression that you could see just with CD209, 206, and 163. And so if we go back and we overlay all this on top of each other, it's sort of the data sort of speaks for itself in terms of both the beauty, but also the challenges in trying to computationally really understand what's going on here if we're to look both at phenotype and morphology. And that is sort of exacerbated by the fact that these data sets then are large. And so this is kind of getting, I guess, circuitously to the point that it ends up being too much data to handle manually. And so one of the things that we focus on is going to be a reoccurring theme I'm going to try to revisit at different parts of this talk, is thinking about as we're moving towards developing automated post-processing and analysis pipelines, what are the main questions that we want to be focusing on in terms of trying to get something that is generalized and extensible that could work on a lot of different tissue studies? And the first one, which is probably where every single study that we do starts, is how do we assign pixels in an image to cells? So when you get the data out, they're just 2D arrays of numbers, right? So we need to somehow be able to get the computer to be able to identify what groups of pixels account for a single cell and to be able to draw those borders. once we have that, then we can start to treat the data a little bit more like you would, say, flow cytometry or mass cytometry or single cell RNA-seq data. We can then extract the cells into a table along with their XY positions and a lot of other things, and we can start to do sort of population discovery based on those cell phenotypes. And then lastly, because this is tissue imaging, ultimately we want to be able to take that information that we found about cell phenotypes and understand how it's all put together spatially. So to start with this sort of fundamental one, we've really spent a lot of time trying to really make some headway on this one. And we've been collaborating with a group at Caltech through the David Van Valen Lab. And this is my grad student, Noah, who has been leading the charge and coming up with a way to be able to have a very extensible general cell segmentation platform. And so this is the next iteration of a machine learning platform called DeepCell, where essentially, instead of having sort of conventional methods for cell segmentation, where you just have a cookbook algorithm that kind of does watershed or looks for areas of high contrast, we actually have trained a neural network to be able to identify cells and images based on manually annotated training data. So the way that works is if you see this cartoon here, say this is a tissue image, you've got like 4 cells. The first step is we need to come up with data to be able to train the network with. And so we take these images and we have humans depicted here as stick people. And those humans are very slow, but they're very accurate. And so we have people go through and actually hand annotate the boundaries of where these cells are. And so now you can see in the annotated image, you get this dark black outline that's going around each one of the cells. So now if we take that annotated image that has the cell borders in it, and we feed that into a network with the original image, we can train a network. We can get enough initial training going to where the network can do an okay job at cell segmentation. And so, and the reason why that we are just trying to get it to be able to do a reasonable job is we can then move on to phase 2 of getting the network where it needs to be, which greatly accelerates how quickly we can get a well-trained network. So now that we have this convolutional neural network that does pretty well on cell segmentation, but not perfect, what we can do is we can now give it new images and ask it to segment those images. And so you can see an example sort of what the output would be like in one of these networks somewhere at phase 2, which is that the model predictions are good, but they're not perfect. So for instance, here we've got 3 cells that the network got right, but then it got one of them, it either missed it or it didn't get the border correct. So what this means though is that in this case, right, we've had the networks actually removed 3/4 of the work for an annotator. So what we then do is we take it back to humans who go in and find the mistakes and fix them. The corrected annotations then go in with the raw images, and we get this nice feedback loop where we now train the network so that it sees where its mistakes were, and we keep going through. And so with each pass of this sort of learning cycle, the network gets better and better and better until we eventually get to the point where it's making pretty minimal errors and it's doing as well as one of our human annotators. And so just to give you an idea of what this is looking like right now, we have, if we have these channels that we're feeding into the network, we've got a channel that shows where the nuclei are. And we also have a channel like, for instance, lamin AC that shows the edge of the nucleus by staying in the nuclear envelope. And then we also have a membrane marker. We put each one of these 3 channels in, and then we ask the network, the first question we ask is like, okay, show us where the nuclear boundaries are. And so you can see that it does really well, even with these even though the cell sizes are kind of highly variable and that the cell nuclei are condensed together. And so that's kind of a blow up. And so, but the thing that we're very happy about is that we've now got this working pretty well in whole cell segmentation as well. And this has been the one that has bedeviled the community for a while. And so Here's an example of that. And again, you can see that it seems to be working fairly accurately on cells of a variety of different sizes and shapes. And so we're pretty optimistic that this not only is going to be working really well for MIBI data, but we've been training this network actually not only on MIBI data, but on a whole multitude of data. And the thing that's great is that it looks like the network training for one imaging modality is pretty readily applied to the other. So we've now got a trained network that can segment data from any of these different multiplex platforms, whether it's Codex or Vectra or SciSif. And the only thing that's required is that you can input into the network a nuclear channel as well as some sort of whole cell marker, whether it's a membrane channel or a high contrast sort of cytoplasmic channels can work as well. And so we're getting ready to hopefully get this deployed and submitted for publication pretty soon, at which point the model will be available to anybody who wants to use it. And so it's actually cloud-based, and it's fully scalable. You do really large images, really small images. So we think we're really excited about how this is gonna really hopefully ease the pain points of doing whole cell segmentation. So now that we've kind of looked at that and we say which pixels in the image belong to each one of the cells, the other two questions we want to think about, again, are cell phenotype, and then how are these cell phenotypes spatially arranged. And so for this second part of the talk, what I just wanted to show you was kind of two examples of studies where that we did, where I think it'll be a good illustration of different ways to approach these problems. And they're actually two fairly, on the surface, seem like fairly different diseases, but that in reality, we found that they've got some strikingly similar functional pathways that are upregulated. And so the first one I'm going to talk about is our first study that we did was on triple negative breast cancer. And then the second one is a more recent one that we've been doing in collaboration with the Gates Foundation, looking at tuberculosis in humans. And so so the first study that we used, both, our first computational pipeline with MIBI analysis on, was this study that was published in 2018 on triple negative breast cancer that Liat Karen led. David was involved in this, as well as Rob West, who's the pathologist that curated this tissue cohort. And so we had surgical resections that were analyzed by MIBI using this 36 protein antibody panel. We ended up getting high dimensional images out. These were segmented, and we assigned cell phenotypes to those. We did different spatial enrichment analyses, particularly with respect to the tumor immune border, and we're actually able to relate a lot of these to 10-year overall survival. So just to kind of give you just a quick summary of some of the findings in that paper was, once we, had the cell segments out, so the first thing that we talked about on the list was we need to say what cell phenotypes are present in significant numbers. And so we can cluster those cells, and this shows the immune cell breakdown in triple negative breast cancer that we found across these 42 patients. And so, you can probably find all of your, favorite immune cell subsets in here. And then if we go and map those back on to the original segmented image, we can pseudo color those cells to actually be able to get sort of a human interpretable view of where these different cell types are. And so if we go back just as a sanity check and look at the actual channel data that those cell classifications were based on, we can be relatively reassured that things look okay. The yellow cell here that's CD20 positive is a B cell. The one cell in here that is orange which is an NK cell, is the one cell that's CD56 positive, so on and so forth. And so now that we've got all of this, we can take this single cell data and look at it in aggregate across the whole cohort. And we see there's what's the first thing that probably jumps out is that there's not only a big diversity of different immune cell types, but there's also, on a per patient level, there's a pretty big variation in what cells are present in which patient and how many immune cells are present in those patients. And so the other thing is that kind of stood out to us initially was that there seemed to be, if you kind of looked at these, a spectrum of the degree that immune cells and tumor cells were sort of admixed together. You know, in some of these tumors, for instance, in like sample 16, it looked like the tumor cells were really just strikingly condensed into these islands of tumor that were surrounded by a pretty cohesive, bed of infiltrating immune cells. Whereas if you look at one, say like sample 32 in this image, the tumor and immune cells look much more admixed and mixed together. And so one of the first questions we asked was, is this just something that our eyes are fooling us on, or can we actually quantify this and get at that? And so what we did was if we take the spatial location of all the cells in that image and we just simply say, are you an immune cell or a tumor cell? And then we look to see to what degree a tumor cell is most often by a tumor or an immune cell, we can come up with a pairwise distance matrix like this on a marker level. And what you see is that essentially immune markers spatially co-localize together more often than not in this particular patient, whereas tumor markers tend to co-localize together as well. But immune markers and tumor markers seem to almost have a preference to be spatially segregated from one another. And this was not the case in every patient. So, for instance, in patient B, you can see that there doesn't really seem to be much of a preferential spatial enrichment pattern. Whereas in A, immune cells seem to coalesce with immune cells, tumor cells, but the two seem to avoid one another. In patient B, it doesn't really seem that anything like that's going on. And so if we look at the pseudo-colored images here, we can see that indeed the one on the left, which we ended up terming a compartmentalized phenotype, there was indeed sort of this striking segregation of immune cells from tumor cells, whereas on the right, they did look much more admixed. And so what this led us to see, at least in this TNBC cohort, was that TNBC seemed to be kind of broken down into two major classes, which have been well appreciated for a very long time before we ever did any of this. The first is cold, which is more or less devoid of immune cells with the exception of, a small numbers of tissue macrophages. The other is ones that are tumor infiltrated. But within that class of infiltrated tumors, what we were seeing was that you could actually break down the tumor infiltrated tumors into sort of two main buckets. One is mixed, which is like we showed, where tumor cells and immune cells are much more dispersed together, and the other is compartmentalized, where they really are sort of preferentially walled off from one another. Now, what was striking was that we then said, okay, so we've got these structural properties that's based purely on, at a pretty basic level, like, how immune cells and tumor cells are spatially organized relative to one another. And we've got kind of two main groups here. Do those two groups, which is purely a structural distinction, is there something about cell function that relates to this structural distinction? And so the first thing we decided to look at was we did PD-L1 in this panel, and we wanted to know how the disposition of PD-L1 expression might trend with whether or not a tumor was compartmentalized and mixed. And so remarkably, we found that there was a very striking breakdown that between whether PD-L1 was expressed on a tumor cell or whether it was expressed on immune cells. And so in this graph, you can see this is a log2 ratio looking at the amount of PD-L1 on tumor versus immune cells. And what we found was that the compartmentalized tumors had far more PD-L1 present on immune cells, whereas the mixed tumors had an overabundance of PD-L1 expression on tumor cells. And this wasn't purely, a ratiometric phenomenon. If you looked at it in raw numbers, indeed, the mixed tumors had far more PD-L1 positive tumor cells than the compartmentalized tumors. And the interesting thing was this finding, which, is a distinction purely based on the structural organization of these tumors, seemed to be coordinated to other aspects of the immune response. And so we saw similar ratiometric relationship with IDO expression, which is the vast majority of IDO expressing cells were found in compartmentalized tumors. And when it was present, it was more often in those compartmentalized tumors to be present on immune cells rather than tumor. But I think probably one of the other striking sort of correlates to go along with the relationship on PD-L1 was that if we looked at PD-1 expression and where it was in terms of whether it was expressed in CD4 cells or CD8 T cells, in compartmentalized tumors, PD-1 expression was far more frequent on CD4s, whereas in mixed tumors, PD-1 expression was far more frequent on CD8s. So that means then that in compartmentalized tumors, where PD-L1 expression was found more frequently in immune cells, that was accompanied by an excess of CD4 PD-1s. And in mixed tumors, where PD-L1 expression was more often on tumor, that was accompanied by an excess of PD-1 on CD8 T cells. So to focus in on the compartmentalized ones just a little bit more closely, the other thing that we found was that the regulatory aspects of this, namely these PD-L1 positive immune cell populations, were heavily enriched near the tumor border. And so we found these populations of myeloid cells that were PD-L1 positive, 11C positive, IDO positive, and that these cells in compartmentalized tumors were particularly enriched along the tumor immune border. And interestingly, that was also accompanied by a gradient of tumor HLA-DR expression, and that gradient being highest right at the tumor immune border, where it's interfacing with these PD-L1 IDO positive myeloid cells and sort of dissipating as it moved away towards the center of these tumor nests. So overall, we kind of put together a very rudimentary, speculative cartoon, and like we like to do in all of our papers, about what sort of the phenomenological findings that we had here. But I think the most striking part was that this distinction of compartmentalized versus mixed was highly predictive of 10-year survival in these patients. So the compartmentalized tumors, those people actually did far better out to 10 years than patients with mixed tumors. And so, we thought that was interesting. There seems to be something here about not just what cell types are there, but how those cell types are spatially arranged, and that those two things aren't independent factors. It seems to be that spatial organization and cell function seem to be sort of inextricably linked, at least in this context. And so, kind of shift to a different subject, and and hopefully the good people at Dana-Farber will bear with me, because this isn't tumors, but I think it has bearing on tumors, which is we're going to talk about human tuberculosis infection, which is a seemingly striking, is a very different disease, but has what we found to be some very strikingly similar features. And so tuberculosis is still a huge problem, especially in the developing world. Around a third of the population, the world's population, has been latently or actively infected with TB at some point. It's still, there's still millions of people that die every year from TB. And so this is a very big emphasis of a lot of public health organizations, and the Gates Foundation in particular, to try to gain some insights into TB pathogenesis. And so TB has been studied for a very long time, and it has a very unique disease pathway, which is that the TB bacilli are usually inhaled as an aerosol. So they're very contagious. It's very communicable through human-human contact, through respiratory droplets. When the patient inhales the bacilli, they're engulfed by alveolar macrophages, and ultimately what that does is it leads to the formation of what's called a granuloma, and so which is a highly structured lesion of immune cells. And so I'll go into that a little bit more detail in just a second. But this whole idea, though, of immune response to TB and cancer is, could they be similar? I'd like to be able to claim that I'm original in thinking about this, but in reality, kind of one of the godfathers of pathology, Rudolf Virchow, had been actually speculated on this probably almost 150 years ago or so, which was looking at TB lesions. He commented that it's interesting that TB granulomas, they kind of look a little bit like tumors. At the same time, though, he said, but they're not quite tumors. And I think what we're about to show kind of reflects those original thoughts that he had so long ago. And so so to kind of like dial in exactly what I mean when I say what is it that we can leverage about one from the other, is that as everybody I'm sure who's working in this field is eminently aware of, key driver of cancer progression as it's been appreciated over the last particularly in the last 10-15 years, but actually known for much longer than that, is that is that cancer progression seems to really require immune evasion in some respect. And that and the other interesting aspect is that this doesn't mean that when tumors progress, you only have cold tumors, right? We have tumors with heavy immune infiltrate that still invade and metastasize. So it's not enough just to get immune cells to a lesion. And interestingly, tuberculosis is kind of, in some ways similar to that, which is that tuberculosis infection progresses despite a very heavy tissue infiltrate. And the other thing that is odd about TB infection that I've always thought is terribly interesting is that if you look at the bacterial load in these lesions with respect to the severity of the disease, there's just not that many organisms in there. I mean, compared to something like, like a bacterial pneumonia, where, I mean, if you tried to culture that, you get tons and tons of, of strep pneumo or Klebsiella or any of those things out. Here, it's like the bacterial load is so low, and the tissue disease is sort of disproportionate for the bacterial load. So it's something very unique going on in TB. And so this is what the lesions look like. And so you can see that almost has this, this structure where the granulomas are here in the bottom left corner. And so this is what I just put an overlay over. This is what's called the myeloid core. So the granuloma is sort of built up with this myeloid core, which is enriched for macrophages, monocytes, and dendritic cells. And this is actually where, and most of those cells are where the organism has been contained and engulfed, but it hasn't been cleared. Now, that myeloid core is surrounded by what's called this lymphocytic cuff, which is mature lymphocytes, which ring the myeloid core, but by and large have a lot of trouble getting into the center. So in TNBC, what we just showed, right, was that the tumor, there seems to be this compartmentalization of tumor and immune cells, whereas in TB, there seems to be a compartmentalization of two immune cell populations for one another. The myeloid core, which is infected with the organism, and then lymphocytes, which are kind of ringing around the outside. And so the question that we had here is that, are there similar cell phenotypes or structural motifs that overlap between these two? If there is there something that we can kind of learn from one by looking at the other? And so the first thing that we found, which we right after starting this, was that this population of immune cells that we found in the compartmentalized lesions, which is here's the tumor immune interface. We've got these, PD-L1, IDO, 11B, 11C positive cells. Well, what's remarkable is we found that in virtually every single TB granuloma we've looked at, the center of the granuloma is enriched with this cell type, which is myeloid cells that express 11B, 11C, PD-L1, and IDO. So virtually every single primary pulmonary TB lesion we've seen, we're getting some of the brightest expression of PD-L1 and IDO that we've seen in virtually any scenario, including tumors. So this got us very interested on this preliminary data, and we engaged Aaron McCaffrey led this, and we, in collaboration with the Gates Foundation, decided to look at sort of a whole cohort of pulmonary and extrapulmonary TB lesions. The workflow is very similar to what we did with the TNBC study, which you can see here. So then going through step one, here's our TB granuloma. We want to go, we've segmented it. Okay, now we know where all the cells are. So if we take those cells and we look at the amount of protein that's in one of those cells, and then map that in. Now we've clustered it. Okay, so we know what our cell populations are. And so now again, we can map those cell populations back onto the image, and we can see where each one of these cell populations are and how they're spatially localized. Now, the thing was that a huge part of TB is that, at least in TB pathogenesis and research, is trying to understand, in particular, the granuloma structure and how does this drive immune cell function. And so we actually, Liat worked with Aaron on this. To be able to answer this question, we developed a new tool for this study. And so we worked with a group at Calico to actually adapt a machine learning technique that's been used for a long time for text analysis called latent derelict allocation. And so essentially what LDA does is it looks through paragraphs of text, and it looks for words that occur repeatedly within close space to one another. So for instance, if you repeatedly saw sort of dog, cat, bear occurring in similar context or in high frequency close to one another, that would be identified as a topic. And so then you could go in and you say, oh, okay, this is a topic of animals. And so we adapted that for tissue analysis by, instead of looking at the distances between words and text, we looked at the distances of cell populations relative to one another in the tissue. And so this is what we call spatial LDA. And essentially what it does is you define like a microenvironment kernel kernel size that'll scan over the tissue. And these studies, we used a 50 micron window, and it goes over and it looks over the tissue image, and it identifies profiles of cell combinations that seem to occur repeatedly in the image. And so just to give you an example then, if we're starting here on the left, this is what we call our cell phenotype map. So here, all the cells are colored by what cell population they belong to. So if we take that and we run it through spatial LDA, what comes out is every single cell in the image gets assigned a probability that it belongs to a different microenvironment. So in this case, we identified 8 microenvironments in TB granuloma. And so in each one of these images, you can see that the opacity of a particular color corresponds to what the probability is that that cell belongs to that microenvironment. So then if for each cell, we pick the color of the highest probability microenvironment, we can now come up with this composite where we can see on the left, the image is colored by cell type. And on the right, each of the cells are now, instead of being colored by what cell population they belong to, they're being colored by what microenvironment they belong to. And so just like with the cell populations, that means we can now state these 8 microenvironments in terms of the frequency that you find specific cell types in them. And so the good thing was we found that very quickly that this was recapitulating sort of what was been classically defined in granuloma structure. So for instance, that lymphocytic cuff that I was talking about, which you can see here ringing the myeloid core in the H&E image, that popped out of this analysis. And so I've highlighted it here. And so you can see that the lymphocytic cuff is going around the myeloid granuloma core. And on the heat map, you can see that that is predominantly made up of CD8s and CD4s, which is what we expect. And so then thinking about the granuloma myeloid core, what came out of that? Well, it turns out that when we did this analysis, the sort of classical myeloid core really was not 1 microenvironment, but 3 microenvironments. And so you can see the sort of relative cell frequencies in here of different myeloid phenotypes that were in each microenvironment. One of them was sort of more BC heavy. The other was enriched for more classical monocytes. And while the last had a lot, also had CD14, 16 monos, neutrophils, and remarkably some Tregs, which I'll come back to in a second. But the interesting thing was that if we go back and we look at PD-L1 and IDO expression, in particular, is there a higher chance that you find these 2 proteins in specific microenvironments not surprisingly, we found that based, on the images, we kind of anticipated this, but we found that PD-L1 and IDO indeed are enriched in these myeloid microenvironments. But we also found that it's not equally enriched across all of them. So you can see that the sort of mCOR1, which is the microenvironment that most closely resembles the sort of classical myeloid core that people have described histologically, has the highest levels of both of these proteins, and followed by the myeloid microenvironment that is populated with intermediate monocytes. So the other sort of powerful aspect of this is, besides just looking at a functional marker and its enrichment in different spatial environments, independent of cell type, is that we can then also ask, if we look at a particular cell population, and so say for instance, in this case, on the left, we're looking at sort of M2-like CD163 macrophages, and on the right, we're looking at 11C positive dendritic cells. And then we can ask, okay, for each of these cell types, what do we see that they have a higher chance of expressing these functional markers when they're present in specific microenvironments over others? And so, for instance, in this case, we found that that was actually the case with CD163 monos. If for those that had when they were present in the myeloid core, and also to some extent in the lymphocytic cuff, more often they would express PD-L1 than when they were found in sort of this other microenvironment called that we term fibrosis. There was a fibrotic microenvironment that was collagen enriched, which is actually where the majority of the CD163 max were. But we found that even though that was the case, even though there were lower numbers in these other regions, that's where, if they would express PD-L1, they most often did. Similarly, we saw some similar stuff with dendritic cells and 11C max as well. The thing about the lymphocyte compartment, probably the most striking thing that we found was, despite the vast majority of CD4s and CD8s being in that lymphocytic cuff region, the exception to this was if we looked at the subset of CD4s that were FOXP3 positive, so regulatory T cells. Those were disproportionately enriched within this myeloid core. And we also found that those cells were actively proliferating much more often than any other T cell population was, once you take into account the frequency of those cells. So here again, we found that there seems to be this structural and functional link that where specific cell phenotypes are doing different things depending on if they're in one microenvironment or the other. Ultimately, we're actually able to corroborate these findings with PD-L1 and granuloma expression in an independent blood transcriptomic study, and found that PD-L1 seems to be a reasonably good biomarker in retrospective transcriptomic data for predicting when patients are going to both progress from a latent infection to an active infection, but also once therapy is started, whether or not they are going to recover from that active infection with therapy. And so we're following up on this right now, because this is actually a big need in terms of trying to identify people with latent infections that are going to progress over time. So that's interesting. So then, we've got this sort of suppressive myeloid population that's found in both scenarios, in TB, and then also in tumors and triple negative breast cancer, which are very different diseases. So is everything the same? No, it's not, which is, also interesting. So the biggest difference that we found, which I thought was really fascinating, was that and when we looked at TNBC, there are a lot of PD-1 positive and LAG3 positive T cells. As a matter of fact, if you looked at the ratio of PD-1 positive T cells to PD-L1 positive immune cells in TNBC, the mean of that log2 ratio was right around 1. So there's a lot of PD-1 positive T cells that accompany the PD-L1 positive immune cells. In TB, it's starkly different. Essentially in TB, you have these bright expressing PD-L1 IDO myeloid cells and virtually no PD-1 positive cells, LAG3 positive cells, or any other marker that would seem to indicate T cell activation or exhaustion. And so this was actually recapitulated also in the transcriptomic data. So here I'm showing a heat map of genes that were, upregulated or downregulated when comparing, in the top row, latent TB to healthy, in the middle row, active TB to latent TB, and in the bottom row, active TB to successful therapy. And the thing that we find was that all of these genes that we're kind of identifying the granuloma, namely CD163, SMA, CD68, all of these, including PD-L1, which is had one of the largest effect sizes, is upregulated during active TB. But remarkably, I think the take-home point with this is that this was in the presence of seeming downregulation of CD3, ICOS, and all these other markers that we just talked about. So this myeloid immune suppression seems to be taking place in the absence of T cell activation. And so, so some of the things that we're thinking about then is, in sort of the summary and wrapping things up here is that, if we do this in this sort of example of what we're calling, comparative immunology in a way, right, is that if we compare two things that are very different but seem to have sort of spatial and phenotypic overlap, what do we see that's similar? So both seem to exhibit these spatially enriched tolerogenic myeloid cells that co-express these markers that have been, sort of associated with almost MDSC-like cells. But the striking difference, again, is that in TNBC, the PD-L1 immune cell expression is definitely accompanied by, phenotypic markers that could be indicative of either T cell activation or exhaustion. But in TB, that seems to not be there at all. And so in TB, this seems to suggest that there's a T cell independent myeloid mediated mechanism of immune suppression that's going on. And so, when I first started talking about this, we benefited from being able to take these findings from cancer, and we went and looked at checkpoint expression in granulomas, and lo and behold, we see really bright expression. And now, kind of what we're thinking about is, can we close that loop? Can we now go back and say, is there evidence for similar mechanisms in tumors? If we go and we look at different tumor types and different scenarios, are we going to find sort of similar situations where there seems to be immune suppressive myeloid populations with a sort of perplexing absence of T cells with phenotypes that would indicate activation? And so, and so this is kind of the stuff moving forward that we're really hoping in the lab to be able to do, which is leverage these diseases that seem to be very different, but look at where they're similar. Because a lot of times it seems that, being able to do that, you're able to really kind of zoom in on some sort of fundamental conceptual process that might be obscured otherwise. So if you want to look at any of this data, we post everything online. The TNBC data set can be found here. We should have, I think we had the TB data set up now. I'll have to go and check. If anybody's interested, wants a job, even in time of COVID, email me. Always happy to talk with people that want to build stuff, do assays, that kind of thing. And again, I'd like to thank all the people that helped us with all this work, particularly David Van Valen and Caltech, as well as all of the really talented people in the lab, without which I don't think any of this would have ever happened. So thank you very much. And if there's any time, I'm happy to take questions.
Okay, thank you. That was a very fascinating talk. I love the images and the TB comparison. So looks like there's no question from the audience, but I have 2 pages of list of questions that I'd like to ask. So maybe I ask some of those. So I read your paper on the triple negative, and if I remember correctly, you used TMAs for those. So it was TMA tissue. So one question is like, what is the size of the tissue that you can image with maybe I know of course you can, but reasonable speed and data in terms of like being able to do it on a cohort? What size tissue? Because in a TMA, they are, pretty small tissues.
Well, so, this is where I feel like I have to be the advocate for the orphan TMA. So tissue micro, so the good thing about TMA is there's good side, there's upsides and downsides. The good thing, so with TMAs, you can actually make TMAs with about any size core that you want. So, the standard, we usually do like 1 millimeter size cores, but you can do 2. I've even seen people who do 4 millimeter size cores. So I think it's the first thing. And the second thing is that, you can make a TMA any way you want. So it's kind of like if you want to take more than 1 core from a block, you can take multiple cores. So I think the important thing about TMAs is to know that it's the real strength of tissue microarrays is that it allows you to be able to build an entire cohort, in a new block, tissue block, which can then be, recut, 50, 100 times, and you can do all kinds of parallel studies specifically right on the same field every single time, which is nice. Now, the other aspect of it, which is nice, is that especially if, like we're in the situation with a lot of times, especially if it's a tumor where you really need to be sure that you're isolating the right areas for analysis, you can have a pathologist go and essentially select the fields of interest to core. And then when you build it into a TMA, it makes it, if you have people in your lab, just purely from a logistics standpoint, like, we have lab staff, which are amazing at these assays, but, aren't experts in the tissue histology for every single disease we look at. And so in terms of like throughput, we've made a lot of improvements in the throughput at this point. We can do about, like one of those fields that I showed in the maternal fetal study, we can complete one of those fields in about 15 to 20 minutes. So it's not, we're at the point where it's definitely still not at the point where you can scan like everything you ever wanted, but it's now at the point to where it's not unreasonable for us to do, cohorts in the hundreds of cores. That's kind of the size studies that we go after now. We're working now to try to improve the speeds, hopefully within the next couple of years by another order of magnitude, so that, you can actually do these much more, something more akin to like a regular, bright field slide scanner. And we can do some of that now at lower resolution. We can actually do survey scans at low res and then go back and look at fields that we picked out and rescan it at high. But by and large, just for ease of use, we find that the tissue microarrays kind of hit the sweet spot of sort of what we can do now. And the other nice thing is that in terms of batch to batch consistency, the good thing about tissue microarrays, you can get like, hundreds of patients on 2-3 slides. And so that means then they can all be stained at the same time, exactly the same way.
No, so I know. I mean, there is a lot of advantages for uniformity. It's just sometimes we've seen that, there is such an extensive heterogeneity, spatial heterogeneity in the tumors. And then the question is what part you take the punch for the TMA. But anyhow, there are a couple of questions from the audience. So question related to how can you segment fibroblasts and macrophages, because the human markers are not well defined for macrophages in particular.
The human, I'd be curious to hear. I mean, we have, using CD68, CD14, HLA-DR, I don't really think we're missing too many macrophages. There may be some tissue macrophage phenotypes that are a little more obscure, but macrophages have actually been, I think, the more, out of everything, have been the more robust staining cell type in a lot of these. In terms of fibroblasts, I think so cells that have more sort of filamentous projections and, and also where you got a lot of ECM around them, that's definitely going to be an area where the cell segmentation can be more difficult. The good news is, though, is that you can use the way that deep cell's set up. You can put in as many single channel markers as you want to assist in the segmentation. So we actually did that in the decidual study. We did a transfer learning model where we incorporated around 5 markers, and we were able to accurately segment a lot of things that wouldn't have otherwise been possible, like these very thin, wispy, smooth muscle layers and stuff like that. So it's the model that'll be deployed, admittingly, is really kind of been trained and optimized for to get 80-90% of use cases. But there's definitely going to be stuff, I have no doubt that when people look at it, they're going to find tissue types where it's like, oh, this isn't performing very well. And so the good news is, though, is that we're deliberately building it out so that you don't have to start from scratch. You can actually use a transfer learning approach to, instead of starting all over, building on top of the existing learning in the model to incorporate new information to assist with that stuff.
Okay. Question related to the deep cell pipeline with traditional watershed. Have you compared the performance of the two?
We have.
Probably
yes.
You did, yeah.
And we'll have pretty extensive comparisons in the paper. Ultimately, if you have a tissue that has very consistent cell morphologies, they can kind of be inferred just on like the nuclear spacing. Watershed might do just fine. Where we found the big thing is the problem that we found with Watershed is that sometimes it works, sometimes it doesn't, and it's not. And one of the biggest problems with cell segmentation, right, is especially when you get into these really big data sets, potentially hundreds of thousands of images, is that if it's not working well enough that you can not have to go through every image, it's a problem, at this point, because it's now getting to the point where you either need to be able to just trust it, or you need to have sort of extracted cell statistics to flag when it's not performing well. And so we're finding that, this is probably the best approach to be able to get some of those things to where, even if it's not performing perfectly, we're thinking we're able to actually build in sort of indicators and flagging functions to say, yeah, this looks like low quality segmentation, it should be inspected.
Okay, question related to the TB, which I found fascinating. So, like you're comparing the active and latent TB, but TB has many stages. Fortunately, we don't see that many cases here in the US, but some people very can progress very rapidly, and also you can have a pulmonary TB or in meningeal, which can be much worse. So have you compared those different stages and different location maybe, and also people who were not treated and progressed?
So the first paper that we have, it's on bioRxiv. The cohort, it's actually two separate sample cohorts. One is around, I'm trying to think, it's probably about 7 or 8 tissues from pulmonary lobectomies from patients in South Africa that had very severe disease. Okay. The ones from Stanford that we had consisted both of pulmonary and extrapulmonary lesions that were really in principle earlier, because these were actually diagnostic biopsies. Now, as you mentioned, and you're totally right that devil is in the details of TB. It can be very tricky because a lot of times the first time people present, it's actually not primary TB, it's reactivation TB, and this is also confounded by the fact that it's very difficult to actually understand what real latent TB looks like, because real latent TB, you probably wouldn't biopsy because, all these, right? So it's sort of, and so, TB, unlike tumors, is a bit more difficult because it's not in any way part of routine care to be biopsying these lesions. And it's only in very severe circumstances they actually excise, tissue. And so, this first cohort we did, we think is really informative, but it's definitely incomplete. And so we're actually following up on that in another effort where we're looking at controlled infections in non-human primates, and we're also building out a new clinical cohort in collaboration with the biobank, ARIA Biobank, where we have around 10 to 15 latent granulomas, as well as ones that were after they've been actively treated. And so that's going to be, and our hope is that we'll be able to take these that have sort of tighter clinical annotation and be able to sort of see how those fall in this controlled trajectory of infection in an NHP model.
Okay. Thank you so much. We have a number of other questions that we won't have time right now to answer. So we will send those to you, and I also can send you my 2 pages of questions.
Sure.
So thank you so much again for this wonderful talk. It's an amazing technology, and we're looking forward to the papers. And thank you everyone for joining us today, and have a great day. Thanks.
Thank you. Bye-bye.
Bye.